right, so thank you everyone for coming to the HPL seminar today. So we have a talk from Baaba Otoo, who's a PhD student here in the HPL. So Baaba started her bachelor's degree in biomedical engineering in Ghana. And then in 2014, she came here and did a master's in mechanical engineering. And then in 2017, she started her PhD with Walter Herzog in the biomedical engineering program. And today she's going to be talking about real-time cartilage cell and tissue strain changes during dynamic loading. So take it away.
All right, thank you. Can everyone hear me okay? Okay, great. So I'm going to be talking about some aspects of my PhD work. So generally, it's titled Dynamic Properties of Chondrocytes. But specifically, we have made attempts to do sort of real-time measurements of cartilage cell and tissue, specifically during cyclic dynamic loading. So now to cartilage 101. When we talk about articular cartilage, because we have other kinds of cartilage in the human body or animals, we are talking about the cartilage that lines the ends of bones of diarthrodial joints. So this might be hips or knees. And as we know, taking the knee, for instance, during your day-to-day activities, it's constantly undergoing forms of loading, which are dynamic in nature. So cartilage is responsible for the transmission of the load, and then also for providing a smooth sort of less friction surface to aid with the movement. So during injury or the onset of diseases like osteoarthritis, you can have some alterations to the structure and the function of the cartilage in the joint. So now taking a closer look at the tissue, cartilage is said to be very unique in the sense that it's aneural, it's avascular, so not connected to any ready blood supply. So when you think about the chondrocytes, which are the only cell type in the tissue, it's quite important when we study the role of the chondrocytes because they've been known to regulate and modify the extracellular matrix and really much just try to keep that homeostatic environment that we have in the tissue. And the matrix is made up mainly of type 2 collagen, proteoglycans like aggrecan, and a lot of water. And when we talk about cartilage, we will typically talk about the different zones because they are also unique in the sense that when you take the cells in each zone, they have different shapes. And then also when you look at the type 2 collagen network, the orientation is also very different in each zone. So it makes the cartilage have quite unique properties when you look at the different zones as it comes. So it's also very important to look at cartilage in terms of the zones. And then thinking of the mechanics, typically we will describe cartilage as a biphasic tissue. So we have a fluid phase and then we have the solid phase. So the fluid phase would be all that water that you have in there with some electrolytes. And then the solid phase would be your collagen and the proteoglycans that you have in there. So the proteoglycans would typically bind to the water because they're negatively charged. So that gives the cartilage, that compressive resilience that you typically see when you try to load it. So in loading cartilage, it's been known that the matrix deforms and the cells deform as well. And uniquely, it's also been shown that the modulus of elasticity is different between the matrix and the cells. So the cells are about 3 orders of magnitude less than that of the matrix. So thinking about the cells and the tissue, there's also a very interesting interplay right there as well. So now let's zoom in closely into the cell or the chondrocytes. And as I said, there's a very distinct, interesting interplay between the chondrocytes and the surrounding extracellular matrix. So when you load cartilage, the cell will sense these mechanical signals and then typically give you a biological response. This biological response might be in the form of something like a calcium signaling, which you can see under the microscope. And because the chondrocyte is connected to the matrix through several links like integrins and other channels, you will typically expect that the loading in the matrix will drive the loading in the chondrocyte as well. So thinking about the mechanical signals being transduced into a biological signal, it has specifically been shown that the changes in the shape and the volume of the cell is what gives us those biological responses that we would see in the tissue. So then when we talk about chondrocyte mechanobiology, mechanotransduction and all that, it's become imperative that we study the changes that are occurring in the shape and the volume at the cellular level as well. Okay, so when we also talk about loading in the cartilage, you will typically talk about static loading and dynamic loading. So with static loading, you apply some load to the cartilage tissue, hold it for a period, let it get to steady state, and then you look at the changes that are occurring in the tissue and in the cell. And so in the past, most of the work that has been done with cartilage mechanobiology has really been only studied in static loading. And it really makes sense because if you think about all the drama that you're going to have with all the motion artifacts, you really want the tissue to get to steady state, and then you could capture the changes that's occurring in the shape and the volume of the cells. But then when we look at our everyday lives, like we are not just standing forever, like we're moving, we're climbing the staircase, we're running, we're taking a walk. And so we really want to know what's happening in a dynamic loading condition, although it might be challenging to capture. And we also really want to know this because it's been established that when you do the static loading configuration, most of the output is of a catabolic effect in the matrix. But then when you are able to induce a dynamic loading condition with the cartilage, you tend to have more of an anabolic response, which keeps that healthy homeostatic environment you want in the cartilage. So then we want to really know what's happening with the dynamic loading conditions. So some limitations that have been there previously that made it difficult to capture these changes during that dynamic form of loading. First of all, as I said, it's because in the dynamic state, you have all these transient changes and not necessarily having the tissue getting to that steady state to be able to capture it. So that has been a challenge in the past. So a couple of studies that have made the attempt to capture the changes during dynamic loading, we're only able to do it at the end of the loading period, which makes sense. But really, we want to know what's happening very early on when the loading starts, whilst the loading is going on, and not necessarily just at the end. Because really, when it comes to the biology of cartilage and everything that's happening in there, yes, certain things like proteins might take longer periods, to synthesize, but then we know that there are some changes that happen in the cartilage that also occur within seconds, right? So we really don't want to miss all that information. And then also there's been challenges with imaging technique restrictions. So really, this is talking more of the technology. So in the past, we've had like great microscopes that could allow you to image stuff quickly, but they also had their limitations. So recently, having a state-of-the-art microscope like the multiphoton we have downstairs, now we can actually collect these 3D images quicker, and then also at resolutions that we can still, not lose information in that sense, because really every pixel and every voxel counts when you want to know changes that are happening in shape and in volume. And then also finally, some equipment in the past had challenges with deep penetration. So most tests would really be more confined to the superficial zone of cartilage. But then we really want to know what's happening across like all zones anyways. So coming up with methods and setups and all that helps us to overcome these limitations. I wouldn't go too much into the methods today since I've done that in previous seminars. So our first objective was to measure the residual tissue strain, and then also the corresponding changes that we have in the volume and the shape of the cells in the superficial zone and in the middle zone during the unloading phase of cyclic loading. And so really what this sought out to do was see how the cells are changing, and then also see what's happening within the tissue in terms of the strains, and then just correlate the two. And then we also wanted to compare and contrast the mechanical deformation of chondrocytes resulting from a static loading configuration and a cyclic loading configuration. So there's a lot of data out there in terms of what's happening in static loading. But because of the new equipment and setup and all that, we just thought to just collect a bit of data also with our own static loading, and then just put all that into context. So that's why we put that in there. So with the hypothesis, in terms of the tissue deformation, we did hypothesize that when the loading starts, the deformation in the tissue is going to be minimal at first, because mind you, there's a lot of water, so then there's a pressure. So when you start loading, the deformation is going to be minimal, but it's going to accumulate through the cycles, and eventually it's going to reach steady state, because as we know, cartilage is poroelastic and viscoelastic. So we need to bear these mechanical properties in mind. And then in terms of the cell deformation, we did hypothesize that it's going to be independent of the tissue deformation, thinking about it being softer and all that. And then also, it's going to be independent in the first few cycles, but then with time, it's going to become consistent with the tissue deformation as it also gets to steady state. So that was a hypothesis for the cell deformation. And then in terms of comparing the static and the dynamic loading, we hypothesized that they're going to behave differently, as has previously been established in literature. So with respect to the methods, so for the sample preparation, we take cartilage specimens from the femoral groove of pigs. These are young pigs. So then we take a 6mm diameter plug and split it into two. So the reason why we chose this configuration of splitting it is so we can turn on the side and pretty much move across the cross-section of the cartilage and then have access to all the zones, as compared to having it, sitting in the regular configuration and just zooming in from the top, and then having the limitation of not being able to go all the way to the bottom. So then that was the configuration. And then we stain this tissue for live cells. And then, so as you can see from the image, these live cells are in green. And then we analyzed cells both in the superficial and in the middle zone, as was mentioned. So for the experimental setup, we have a custom-made cartilage compression device. And so with this device, we place the semi-cylindrical cartilage plug in here. And this whole bath is filled with phosphate buffered solution. And so over here, we have the compression plate, and then we have a motor that picks the signals from the displacement sensor, because this is done in displacement mode configuration. And so with this setup, the reason why we would term it real time is that we place all of this on the stage of the multi-photon microscope. And so then we have the microscope objective here. So as the loading of the cartilage is taking place, we are intermittently imaging and collecting Z stacks of the cells of interest, which we follow all throughout the protocol. So this is the experimental setup. So in terms of the loading protocol, for the static loading, we chose to apply a 20% strain and held it for 20 minutes. And so we collect images of cells at a reference point at time 0 before applying the load. And then over time, we collected at 5 minutes, 10 minutes, 15, and then 20 minutes to give us a data set. So now of much more interest is the cyclic loading. So for the cyclic loading, again, we applied the 20% strain magnitude, and this 20% is within a physiological range when you talk about cartilage loading. And so this load was applied at a frequency of 0.2 hertz, and we did a total of 100 cycles. So again, we collect the images or the Z stack at time 0, and then we chose different time points all throughout the 100 cycles. So if you observe, we collected more images during the first 10 cycles because we believe there was a lot going to happen within the first few cycles. That's going to be very important to us. So we didn't really want to miss out any information. And then subsequently, we collected at longer time periods. Obviously, we would have loved to do more imaging time points, but when it comes to imaging, you want to consider photobleaching, the effects of photobleaching over time. So as I said, every pixel, every voxel counts. So you don't want to trade any of that. Yeah, so then we did do some reliability tests, and this number of time points was still good enough to be used. Okay, so with a cyclic loading, this is what we have. So then we're going to have all these multiple image stacks of the same group of cells that are followed all throughout the 100 cycles. Yeah. So once we have our Z stack, we then ran it through an open source package, which was developed by Scott Sybil. So with PyCell, first of all, we are able to segment the cells 3D. And then what happens then is that we can now go ahead to do the kinematic calculations. So remember, we have our reference stack images, and then now we have the images at the different time points. So then we can compare the images at the different time points to the reference to see what the changes are in terms of the strain. And so we also define the height of the cell. So PyCell assumes the cell is an ellipsoid. So then I guess putting that into context, you can think about how the calculations might work a little bit easier. So then the height of the cell is defined as the direction in which the load is applied. And then the width is going to be in, I guess, can I say opposite direction? Yeah, and then the Z is going to be the direction of our laser where the imaging was done to collect the different stacks. So then now we have our segmentation done for the different time points, and then we can do the math to see what the differences were by comparing it to the reference. So going further, we wanted to know what was going on in the tissue in terms of the strain, especially because we collected these Z stack images at the unloading phase of the imaging. So of course, to start, that was the easiest approach because then again, motion artifacts. So then you want to collect it after applying the load releasing the load, that time period, that was when we collected these Z-stack images. So we called those at the unloading phase of the loading. Yes. So then because we're collecting at the unloading phase, we wanted to know if the tissue was still in compression during that unloading phase, or pretty much how much recovery had happened during those few seconds when the load was removed. And when we think about cyclic loading, so as I mentioned, we have the surface of the cartilage, and then we have the compression plate that's loading it. So you would really imagine that over time, there's going to be a point where it's not going to be in contact because you're loading for 100 cycles over time. Yes. And yes, this is a slow form of loading because with the 0.2 hertz, that meant 5 seconds to complete one loading cycle. So then we're going to have times when they are not touching, as I said. So then we just thought, why don't we use that lift off as we describe it to give us information of how much the tissue was compressed during those unloading spots that we took the imaging of the cells. So then we came up with a MATLAB code to help us do this. So if you look closely at this, the loading device gives us information from the displacement sensor, as can be seen at the top. And then we also have information from the load cell for the force. So then we defined a baseline for the force. It couldn't be 0 newtons because when we were starting, we had to give a little bit of a tower load. So then let's say about 0.2 or 0.3 newtons would now be our new baseline. So then we defined the lift-off distance as the difference. Okay, so if we look at the displacement, looking at the blue triangle at the top, right? So then we defined that as when the force gets to the baseline and you trace it upwards and get to the point of the blue triangle, that's the difference between the spot and the baseline of the displacement was going to tell us that the compression plate was not touching the tissue at that time because force has pretty much come to 0. But then the displacement is not necessarily at 0, but then whenever the force comes to 0, that means that there's no, load applied to the tissue at the time. So just using this ideology, we were able to do the math and then estimate our lift-off distances at the unloading phases where the imaging was done. And then we had more information on how the tissue was behaving. So, sorry. So now this is a graph I put together from the force response and the displacement response, or in this case, the lift-off response. So we have force on the left, lift-off on the right. And so looking at the force, so just starting from the very first cycle, we have a higher force because obviously there's that pressure buildup. And so at this point, the fluid phase of the tissue is pretty much carrying the load. So we have a high force here. And then as we go down, you see that typical viscoelastic behavior curves that you would expect. So we see the force rise again at various time points because these were time points right after imaging was done because we had to add a few extra seconds to that 5 seconds at the bottom just to be able to capture the entire Z stack that we wanted. So there was a little bit of a recovery in that sense. So that is why you see the force go up at the different time points that we had imaged. So now coming to the lift off, as we can see, we saw a decrease in the lift off when the force was higher as expected. So then we now have information that lets us know that the cartilage was still in compression, even though we were imaging at the unloading phase when there was no force being applied. So this became very important information, as we will see very soon in the results. So yeah, we were happy about the liftoff thingy. Yeah. Okay, so now looking at the results, just as I said, so with a liftoff distance, we are now able to calculate the residual tissue strain during the unloading phase. So if you remember, the nominal strain that was applied to the tissue was 20%. And this data for the strain from the liftoff, again, was obtained at the unloading phase during the loading. So as we can see from the very first cycle, we have less strain because we don't have much of a gap or space between the plate and the tissue. And over time, we see more of a compressive strain still in the tissue, even though we were at the unloading phase. So this strain information becomes very important because now when we move on to the results we got for how the cells were changing during the cyclic loading. So for the superficial zone, interestingly, what we observed was that the cell first loses volume within the first few cycles, but then surprisingly, the volume recovered over time and pretty much tried to get back to baseline volume. But then remember, we did notice that the tissue was still compressed. So although the tissue was in compression, the cell still managed to recover its volume as the loading was still taking place. And so this was very interesting to us as well. So the whiskers represent the maximum and minimum values, and then the lines represent the median. So again, in the superficial zone, for the very first cycle, there wasn't much of a volume change. Again, that pressure buildup with the very first load applied, with the fluid phase taking much of the load because of the hydrostatic pressure buildup. So then this was a very interesting phenomenon to see in the superficial zone. And then with the middle zone, pretty much not much happened. The volume of the cells pretty much remained constant all throughout the 100 cycles. Again, remember, when we talk of cartilage, each zone is unique. The cells are shaped differently. The collagen fiber orientation is different in each zone, so they all behave quite differently. So this was what we observed comparing the superficial and the middle zone. Now, in terms of the cell morphology, thinking about the shape, we went ahead to look at what was happening with the height, the width, and the depth as well. And so when we look at all of them in total, they pretty much behave kind of how the volume was behaving, with the largest deformation being in the height, which was the direction of the load. So then we see again within the first few cycles, so you have a decrease in the height, the width, and the depth. So you think of it more of like the cell shrunk in all directions. And then over time, we see the recovery of the shape just as we saw of the volume. And then again, with the middle zone, not much activity happening in terms of the shape changes as well. So then now looking at comparing the static to the cyclic loading, as expected, the cells for the tissues where the static loading was applied lost a lot more volume than the cells during the cyclic loading, which we kind of expected. We did the comparison at the 5 and the 10-minute time points, because remember, for the static loading, we didn't have multiple time points like the cyclic loading. So we chose to compare them at 5 and 10 minutes. So for the cyclic loading, the 5 minute mark was pretty much around the 50th cycle, and then the 10-minute was really at the 100th cycle. And so pretty much the cells were recovering their volume at that time anyways. So then when you look at this, you now begin to wonder, okay, this was collected at the unloading phase. So what's really happening at the top of the loading, which I will get to shortly. And another interesting phenomenon also was in comparing the shape changes between the static and the cyclic. We saw very distinct differences where in the static loading, you have that decrease in height, but then you have that expansion in the width, which has previously been shown in literature. But then again, when you look closely at the dynamic loading, its shape changes were unique in the sense that it almost feels again like it's really shrinking because it's decreasing in all directions. So those were the two distinct differences between the static loading and the dynamic loading. Yeah. So then we now get to the point where we ask ourselves, so we're collecting orders at the unloading phase. Yes, it was a challenge initially. We managed to overcome it, but really it's easier when you think about it and with respect to what's happening at the top of the loading. So we had to come up with ways to be able to image at the top of the loading, although it's as challenging as it can be, because then again, there's going to be so much motion artifacts because you still want to keep the loading configuration as dynamic as possible. And then you end up defeating the purpose if you really get to the top and hold it for a long period of time. And then you only image after it's gotten to steady state. So the next objective was now to measure the changes at the top of the loading, as I mentioned. And this time around, we're really pretty much just interested in superficial zone because obviously the middle zone has shown us I'm chill. So just think about the superficial zone. And really, if you think about it, even in diseases like osteoarthritis, it's usually the superficial zone that's mostly implicated anyways, right? So we really want to know what's happening more there. So we just focus on the superficial zone for now. So then we now came up with a new protocol. Well, not new in that sense. Same loading protocol, but new imaging protocol. So then we now, after getting our reference stack, Z stack for the reference, we now picked several spots at the top to image and then still at the bottom, because then really, if you just do the top, you really want to know what's happening collectively, right? So it didn't really make sense to just only focus on the top, but sort of to disperse it this way to see what kind of information we can now get. So again, now, so we have Z-stack images at the top and Z-stack images at the bottom. In order to be able to achieve this, I won't lie, it was really challenging, but over time, we were able to come up with ways. For instance, it took me several weeks in the lab to realize I could play around with the PID controller to control the oscillations, because there was so much oscillations that was happening at the top. And then also, just around that time, Scott came up with an improvement to the code. So then now it was more improved, even with the resonance imaging, to be able to segment the cells in sort of like, even though having like way less resolution in that sense. Because remember, we trade the resolution to have less time to be able to image quicker. So then we're able to come up with a way to get this going. So now for the results. Initially, we thought, oh, yeah, you load at the top, since it's under compression, the cell is going to be smaller. So maybe we're going to see a smaller volume at the top, even compared to what we imaged at the bottom. But surprisingly, just looking at the top, we actually realized we had more of a volume increase. And mind you, these are not middle zone cells. These are superficial zone cells. So that was really surprising to see a volume, well, a bit of a volume increase, just focusing on the bars of the images that were collected at the top. Again, we still saw that behavior where you have a bit over time, you have a bit of volume decrease within the first few cycles. And this time around, it took a while. It actually got to 20 cycles. The first protocol, we saw that volume increase within the first 10 cycles, but this one, it was still within the first 20 cycles. And then we saw that recovery. If you look at the X, so again, the line is the median and the X is the mean. So then we still saw the volume recovery up until the 100 cycles, which was comforting for me because then it did show that the results were reproducible, like in that sense. So now looking at this behavior, at the peak of loading, we had a volume increase. And then when the load was removed, then you have the cell shrink. And then you apply the load image again at the peak, you have a bit of a volume increase, even though over time that volume increase became less and also went to its own sort of recovery of trying to stay at that baseline volume that you started with. So then this became interesting, like, okay, so what's going on? The load is applied, but the cell is still managing to gain a bit of volume. So that was very interesting and weird, but digging through literature gave us lots of explanation, which I will talk about during the discussion. So now looking at the shape behavior with this one as well. So first looking at the width, we had the pattern of the width also being similar to that of the volume. And then with the depth as well, even though the depth was a bit more or less, you still have that sort of increase, decrease behavior. But interestingly, when we got to the height, the cell seemed to be keeping its height, keeping it intact over time. And that was very interesting, considering the fact that that was the direction that the load was applied. But if you think about it, it really could be an adaptive feature because when you think about cells and with loading, we know that the cell membrane cannot be stretched beyond a certain limit, which is about 4%, right? So then looking at the direction of the load and the height being the direction of the load, if it was really going through so much changes, then you begin to think about maybe rupture due to fatigue over time. And yeah, so those are the kind of thoughts that I have in my mind when I look at the cell trying to keep its height as stable as possible. So now, so just to discuss again, from the very first results we saw with the cell, in terms of the superficial zone, we saw that volume loss and then the recovery. And when you look through literature, not just with chondrocytes, but even with other cells, it's been shown that cells lose volume as a protective strategy. And during this volume loss, the cytoskeleton in the cell actually remodels to allow the cell to be able to sort of take on the load that's coming in. And it's been shown to be a good adaptive feature against impact loading, for instance. So you tend to see quite a bit of this happening during protocols that are more of like impact loading in that sense. So volume loss has been shown to be a protective strategy for cells. And then volume recovery also. So with the volume recovery, yes, when we talk of cartilage, we say, oh, the matrix is passive, but we really don't need to think of the cells as being passive. We need to think of the cells as being active. Like, as I would say, they have a mind of their own in that sense. So if you think about the volume recovery in that sense, for sure, there's active regulatory processes that's going on. in the cell that enables the cell to try to maintain its volume as much as possible. And we see this behavior also during loading configurations like osmotic loading as well. So it's been shown that there's a bit of volume recovery in there as well. And then when we think about that very first cycle, well, the first few cycles where we pretty much had that volume change. So it's also been shown that during processes like calcium signaling, right? So when you begin to load the cartilage underneath the microscope and you see the calcium signals as these sparks, you only see them within the first few loading bouts, and then you don't see them again. So then with graph looking that way, where we lose the volume and we have the recovery, it kind of makes sense when you think about it in terms of biosignaling or whatever is happening biologically in the cell, telling us that there's a lot happening within the first few cycles before the loading accumulates and works its way towards steady state. So that is also sort of, yeah, throwing more light on the data that we have. And then with what was happening in the middle zone, I would say kind of expected, because the middle zone cells deform minimally, and it's been shown that there's a lot of difference between the stiffness between the middle zone and the superficial zone. So if you look at it, the middle zone tends to be a bit more stiffer. I mean, it has more proteoglycans in there, so it's going to bind to more water. So you're going to have more of that compressive resilience. So it will make sense why the middle zone cells will not deform that much. And then again, yes, as I mentioned, you have the hydrostatic pressure in the middle zone being higher. So then the cell is able to maintain its shape and volume. And then finally, also, it's been shown recently that the pericellular matrix, which is just right around the cell, also actually is quite different between the superficial zone and the middle zone. So then there's a little bit more of a mechanical protection for the cells in the middle zone compared to the cells in the superficial zone. And this pericellular matrix is also quite rich in collagen 6. So then thinking about the fibers in there, obviously mechanically, it's going to be, contributes more to like a more protective function around the cell. Okay, so now talking about physiological conditions that involve a change in cell volume as a sensor. In the beginning, I had spoken about how these mechanical signals are transduced to biological signals. And it's been shown that one of the, or most importantly, it is the changes in the shape and volume that is contributing to us seeing these biological processes. So then if you having a normal cell, if you then have a cell that has an increased volume, when you go through literature, you can see that increased volume has actually been linked to what we will call anabolism in terms of, so I call anabolism good metabolism. Yeah, so then if you think about having the cells being larger at the peak of the loading, even though we weren't expecting that, we know with dynamic loading, it incites a lot of anabolic processes in the cell or in the tissue. So then really, if we have an increased cell volume, then it will make sense because we know there's a lot of anabolism that comes with dynamic loading. And then also when you think about other physiological processes like absorption and stimulation, and even cell proliferation and cell migration. And well, also in necrosis, it's been shown that you have a bit of an increase in the cell size before that also happens. But really, a lot of the good stuff seems to happen with the increased cell volume as compared to the decreased cell volume, where when you dig through literature, you see more of a catabolic effect with a smaller cell. And then also, there's also the programmed cell death that also happens. Well, a decreased cell volume precedes programmed cell death Yeah. And so you have other processes also like cell differentiation also happening with a smaller cell size. So in conclusion, we were able to show that the static and dynamic loading protocols, when you consider a similar nominal tissue deformation, which was 20% in the sense, and then the time period, it produces completely different changes to the cell volume and the cell shape. So this has been established. And then secondly, the chondrocytes in the superficial zone experience vastly different cell deformations and the recovery during cyclic loading than the middle zone cells. So there's that distinct difference between the behavior of the cells in the superficial and the middle zone. So I would like to thank my supervisory committee, Walter, Dave, and Neil, as well as all the people in the Herzog group that have been of great help to me. And then finally, this is a summary figure. So again, we have a setup for simultaneous loading and imaging. We sought to look at what was happening during cyclic loading. And then with the tissue response, we see that compressive behavior, even though it's at the unloading phase. But then with the cell, we saw the dip and then recovery over time, even though the tissue was still compressed. And then when we looked at what was happening at peak versus the valley or the unloading phase, we saw that increase cell volume at the peak, but then also eventually getting to baseline. So that's it for today, and thank you.
Thanks, Bubba, for your awesome presentation. Go to slide 9.
Sorry, I have to go the old school way. Okay, 9.
No, I guess a few slides further. The first time you're showing that loading cycle.
Oh,
Next slide. Next slide. Next slide. Yeah.
which was on cyclic? Oh, okay, static.
Yeah. So you applied strain,
Mm-hmm.
and then you kept it constant for like some minutes.
Yeah.
And is this just one cycle?
Yeah.
And so does that mean you captured kind of like a relaxation response?
Yes, we do.
And then did you also monitor the changes in the relaxation responses you got between different cycles?
Yes. So I'm not sure if I have that graph here, but so looking at the force curves between the static and the cyclic, there was also a couple of differences. For instance, there was a higher force response in the static than in the cyclic when you compare the values. And then also the force at which the tissue gets to steady state in the static and the cyclic were also different. So I'm not quite sure the information you want over there.
Okay, sounds good.
Yeah.
And then my other question is, did you give cartilage enough recovery time between each loading cycle?
For the cyclic?
Yeah, for example, between cycle 1 and cycle 2, cycle 2, cycle 3.
No, because then that defeats our purpose of real time, right? Yeah. So.
And then my other question is, why did you use explants from femoral groove instead of tibial plateau or femoral contact?
Okay, so tibial plateau, 2 cents. Yeah, not a personal favorite. Yeah, also because again, so these are younger pigs, right?
Yeah.
So you don't have that distinct differences in the different zones. So then working with thicker cartilage was helpful in the sense that we could now define what our superficial zone would be. So our superficial zone was the first 100 microns, and then our middle zone would be 100 to 500 microns. So that was sort of the area that we worked in. But then also by observation, if I saw more spherical cells, then I'm more convinced I'm in the middle zone as well, as compared to the superficial zone that was more flattened. So these tissue specimens were approximately 2,000 microns thick, or 2 millimeters. So yeah, so again, tibial plateau too thin, femoral condyle too round. So then I don't want to be resolving for forces and all that at the surface. So then the femoral groove gives you that perfect flat surface, so that when the indenture touches, we know that we can estimate the force just as it is.
Yeah, I understand that it's challenging.
Yeah.
In terms of the thickness and high curvature,
Mhm.
for instance, in the femoral condyle. But the question is, are your results applicable to the femoral condyle? Because your results are based on femoral.
Yeah, I mean, that's really a good point, because cartilage behavior in terms of the biology has somewhat been shown to be region specific. So then some regions are more loaded than others. And some regions experience more shear than others. Some regions would get pretty much your typical compression and less shear. So for sure, the cells in those portions of the tissue might have adapted in some way, but this is more a proof of concept sort of study to just give us initial information as to how the cells are behaving. So it's safe to use a femoral groove. It's straight, less stress. And yeah, I think that's what I can say for now.
Okay, awesome.
Yeah.
Thank you.
Thank you.
Oh, thanks, Baba. That was a great talk. So I'm interested to understand more about the time varying change in the volume of the cells during the static load.
Mhm.
So did it have a similar recovery as dynamic, or it's almost was the same as the tissue response, where it dropped and then it just plateaued
Exactly. Yeah.
at that compressed? Okay.
Yeah. So, but that has been shown previously
Yeah.
several times, so we can expect that.
And just to follow up, do you think that under a higher loaded magnitude, because I understand this is like more of a moderate,
Yes.
do you think it would mimic the dynamic response or the static response that you saw?
So let's say I went above 40%.
Yeah.
I don't know if the cells would be, they might try to recover,
Or you think it would
but
almost
probably
mimic static?
wouldn't get to that point. That's what I think.
Great.
Yeah.
Thanks. Yeah, please.
So you were talking about the difference between superficial and middle zone, and so did you look how I know you did a strain, nominal strain, but did you measure how the strain changes between the zones and in the region where the cells are?
So that's
Like
what I'm,
around the cells?
so that's what I'm working on now. So just give me a few
Yeah.
weeks.
Okay.
Yeah. So that's my last research question. And so that's when the grid comes in. So obviously, with this information that we got from the force response and then using the MATLAB code, yes, it's telling us but it's more of a bulk strain, but we really want to know what's happening locally. And then we also want to know how the strain is varying also in 3D, just like the cells. So that's why we would imprint the grid and then load and then see how the tissue matrix next to the cell is also deforming. Yes.
So do you have any speculation? Like, do you think that there are different local strains that are responsible for some of these differences?
So for sure, the matrix is not going to recover like the cells, that I know for sure. So for this test, personally, I am actually just more interested in looking at the strain distribution over the cross section. Yes, that has actually been done. Amin has a paper on that, but what would make this current one unique is it's going to be in 3D. So then not just in one direction. So that's just going to be like a little bit more, well, a little bit more added knowledge to science. Yeah.
What did Amin's work show about the heterogeneity of the strain as a function of that?
Yeah, so as a function of that, for sure, the strain changed as you went towards the bone.
Yeah.
So, but then,
Less.
yeah, but then the thing again is one key limitation with this is the young pigs that we're using. Yeah, the cartilage is thicker, not quite defined in that sense. So it would have been awesome to get adult pigs to use for that, because for one, you have that distinct, the distinct zones that you want. And then also, I feel like you will see better distinct changes because I find these young pigs, I don't know, in my head, I feel like the cartilage has more water. Yeah.
But would the strain be heterogeneous like that even in a static loading case, or does under static loading does fluid get redistributed and it just kind of ends up being quite homogeneous? That's a question. I said it like it wasn't, but
Yeah, you still see that.
you do.
Yeah,
That's really interesting.
yeah, you still see because.
And especially if like you're not getting deformation in the deep and middle zones of the cells, it begs the question, why do you have cells there anyways? I'm not explaining it. I always thought when you do cyclic loading of cartilage, I always thought that at some point it would just start looking like a long creep test. Like if you didn't allow enough time, you would just have this fluid and it would exude and it would exude and it would exude, and then at some point it doesn't matter, you would just be loading the matrix or whatever. And so I was just wondering, like, what would your data look like if you loaded it like 2 hertz or 3 hertz or something that mimicked kind of like running? Yeah.
So I have thought about that. I would have loved to do that, but the current the controller we have in the device is not going to allow us to do that. Yeah. So I've actually not thought about only increasing the frequency, but also increasing the duration of the tests as well. And for sure, it's going to get to that point where, yeah, no,
You think
it just becomes,
it does kind of look, at least from the graphs that you were showing, but the force data did kind of, it did look like it plateaued really quickly.
Yeah.
Like after 10 cycles.
Yeah. Yeah, absolutely, it did. So I still think in that context that 100 cycles is a good amount of time to give us the information that we need. But really, when I think about that dip and the recovery, I personally, through intuition, believe that over time, there's going to be a point where it's going to try to dip again, but it's not going to go as low as the previous one. And then you're going to have the recovery back to baseline. And then over time, that dip will pretty much become non-existent in that sense. So that's sort of how I think about it. Yeah, but it all eventually just gets to steady state.
But I do have a question about where the fluid comes from. Is it all pericellular? Is it extracellular? Like, how do you think it sort of increases its volume like that?
Yeah. So yes, definitely from the ECM, the ion channels. So the connection has been distinctively shown. So then you just have that change in potential, and then it just does its thing between the ion channels. Oh, and then there's all these aquaporins, and I still need to brush up on my biology.
And then that issue where in static loading, you kind of see this Poisson's effect where it kind of expands in the horizontal direction. Are you sure you don't see that in the dynamic case because you're not looking at it at peak loading? Or when you were able to look at it subsequent study did you also did see?
A little bit, but not as much as that.
Not as much as the
Yes.
static case.
Exactly. yeah.
Okay. So it does do that.
It does do that, yeah. But I actually choose to see imaging at the peak as the first few seconds of your typical static loading,
Yeah.
right? Yeah.
Any last question? Venus?
I'm not gonna like pressure you on biology, but you had on your slide the decrease in the cell volume will lead to the catabolic effect, degeneration, and then you had cell differentiation. So did you mean that the cells would change, the chondrocytes would change into another type of cell?
So I'm looking at it, so digging through literature. So I was just looking at different physiological phenomena where you have smaller cells. So that before the differentiation occurs, then the cell gets smaller. So not necessarily the chondrocyte changing into something else per se, but just at a higher level. So that last slide was actually talking generally about cells, not just chondrocytes. So because I was just trying to make meaning of, okay, if a cell is increasing volume, what's happening there? Is it good? Is it bad? And all that. So that's not just for chondrocytes, but cells in general. Yeah. Sometimes when you try to find information for only chondrocytes, you don't find much. Yeah.
Just curious, what loading configuration did you use unconfined compression?
Yes, to an extent. So the cartilage was free all around and only covered at the top, confined with the cover slip for the
So
objective
that
lens.
indenture that you compressed cartilage, was it permeable? Or
No, it's not. It's not permeable.
the whole sample was immersed in PPS?
Yeah. Oh, PPS. Yeah. Yeah, because the objective lens needs to be in a fluid. So, anyways. Yeah.
Thank you so much, Baba. Let's give her
All
a
right.
hand.